FMD is endemic in Pakistan, and 3 serotypes, that is serotype O, A, and Asia1, are in circulation. And there are multiple subtypes within each serotypes. Studies on genetic diversities of FMD in Pakistan, in different areas in Pakistan, are already available. However, similar information about FMD in Balochistan province of Pakistan is not available. So the objective of this study was to characterize FMDVs originated from Balochistan province of Pakistan. Balochistan is located in the southwestern part of the country. It is the largest province of the country that shares borders with Afghanistan and Iran. The total border between Pakistan and Afghanistan is about 2,430 kilometers, of which Balochistan shares 1,100 kilometer border with Afghanistan, and with Iran, Afghanistan or the Pakistan share about 909 kilometer border. The geographical location of this province has importance in animal trade, both formal as well as informal, between Pakistan, Iran, and Afghanistan. And this formal and informal animal movement or trade results in spread of various transboundary animal diseases, including FMD. In the study, a total of 35 samples were collected, 2 epithelial samples and 33 oral swab samples. These samples were collected from clinically suspect cases of FMD during April 2011 to November 2011, and these originated from 8 different locations within Balochistan province. These samples were transported to ANSES Laboratory for Animal Health in France, and these were further analyzed there. The samples were subjected to antigen ELISA and also cell culture and subsequent antigen ELISA. These samples were also tested using pan-FMDV real-time 3D assay and serotype-specific real-time PCR assays. And then VP1 sequences were also generated from some selected samples. All the samples, the original samples, were tested negative in antigen ELISA. However, 8 of them virus was obtained using tissue culture, of which serotype A was found in 5 samples, serotype Asia1 in 2 samples, and there was 1 sample showing positivity for both serotype A and Asia1. Out of 35 samples, 31 were positive in pan-FMDV 3D assays, and 29 samples were positive for serotype-specific real-time RT-PCR assays, which included 16 serotype A viruses, 8 Asia1 viruses, and 5 samples showing infection of serotype A and Asia1. To our surprise, no sample was positive for serotype O virus. However, this may be due to immunity due to previous infection of serotype O or due to vaccination. There is some issue with vaccine matching for serotype A and Asia1, so that may be the reason that the samples were positive for serotype A and Asia1 but negative for serotype serotype O. The serotype A viruses, we found 3 different sublineages within serotype A, and they all belong to A-Iran-05 lineage. The 2 sublineages were the previously described FAR11 and HER10 sublineage, and and a new sublineage which we named here as BAL11 sublineage. The FAR11 sublineage viruses were very closely related to the viruses circulating in Pakistan and Afghanistan during 2009, whereas the HER10 sublineage viruses were very closely related to the viruses circulating in Afghanistan in 2010, in Kyrgyzstan in 2011, Iran in 2012, and Kazakhstan in 2012. The new sublineage, we didn't find a close relative of this sublineage while during blast. For serotype Asia1, all the viruses, they belong to group 7 of serotype Asia1, which is also named as Sinzur 8. And this lineage was, our group was first detected in Pakistan in December 2008, and later in Sindh province, that is the southern part of the country, and then it spread to other parts of the country, and then it was found circulating in Afghanistan, Iran, in the Middle East, and then it was detected in 2011 in Turkey. So the viruses belonging to Asia 1 belong to this sublineage group. So in conclusion, different sublineages within A105 are in circulation, and new sublineages arises with time. And our previous study suggests that it takes 3 years for serotype A105 lineage to arise a new sublineage within this lineage. And detection of new strains of serotype A suggests that continuous surveillance of the disease is necessary for the area. Oral swab sample can be used for virus isolation and subsequent characterization. And collection of oral swab sample is non-invasive compared to the epithelial samples and are therefore acceptable to the farmer or animal owners. Detection of two different serotypes or two different strains of the same serotype in one animal or sample shows complexity of the disease in the region. I would like to thank Asadullah, belonging to Balochistan, who collected the samples, and his team, and Stefan Zintara and Labib and their team for analysis of these samples in their labs, and also the EuFMD team for their interest in my participation in the meeting, the Higher Education Commission of Pakistan, who sponsored these studies, and GFRA, who funded my travel grant for this meeting. Thank you.
Thank you, Jamal, for this nice presentation. Any question? Yes, please.
Were those samples collected as a result of passive surveillance or active surveillance? In other words, are they collected as reported cases of disease, or you have made some active search on the field to see what is the situation?
Actually, the disease reporting system in Pakistan, and specifically in this part of the country, is non-existent. So these were collected upon report that there is some disease similar to FMD. So that was part of that activity in which they were collected the samples. But there is no regular reporting system of the disease in the whole country, and specifically in this part of the country.
Thank you very much for the presentation. I didn't understand why there was no virus serotype O found in any of those samples.
Yeah, this is, to our surprise, no sample was positive for serotype O, but this may be due to the fact that there may have been some previous infection of serotype O, and due to immunity, there may be not current circulation of serotype O virus, or due to previous vaccination. But for serotype A and Asia1, as there is problem in vaccine matching specifically with the current circulating serotype A and Asia1 viruses. So that may be the reason.
Question.
Okay, thank you very much. One question about these double viruses. In a number of samples you detected two serotypes, and if I understood it correctly, you took oral swabs. So could it be that a double serotype is caused because of a clinical disease in a carrier? So that one virus is from carrier status and the other one is actually representing the clinical disease. And can you conclude that from the CT values, for example, that one of the samples would have, or one of the serotypes would have a high CT value and the other CT value would be much lower?
Yeah, well, as far as the sequencing are concerned, we sequence only those samples having low CT values, that is 32 or below. And there is a chance that the samples may have been contaminated during collection. However, if we see if we compare the different sublineages of serotype A and different branching pattern of serotype Asia 1, then this possibility is very, very minimum, very, very low.
Any other question? I have just one related to surveillance in some countries that difficult to do sampling.
Yeah.
Can you say some words about how Assad went to the field to collect the samples, how it was difficult to collect samples in this area?
Yeah.
Please.
Yeah, actually, the situation in Balochistan is not very good in terms of security. And he actually tried to contact some friends and ask them to inform him about FMD outbreak if there is any. And in this way, when an outbreak was reported to him by his friend, then he went there and he collected the samples.
Okay, thank you very much.
Thank you.
The next presentation